Bismillah, Assalamualaikum warahmatullah. You're listening to immunology lesson on immunity to bacteria, viruses and parasites. By the end of this lesson, your learning outcome is you can describe mechanistically how immune system deals with bacteria, viruses and parasites. So why these three? Because bacteria, viruses, and parasites are the most common infectious agents you'll encounter both clinically and in daily life. So if you understand these three, you have solid foundational blocks later, especially if you want to study less common infections or pursue advanced research. As an overview, I want you to think of two key ideas. The first key idea is our immune system responds differently depending on the type of microbe, whether it's bacteria, viruses, or parasites. And the second idea is sometimes the damage in an infection comes from our immune response more than the microbes themselves. So our immune system, which protects us, can also unintentionally harm us. So we'll look into both of these ideas as we go through each type of pathogen. Now, let's start with our first type of pathogen, extracellular bacteria. So what does extracellular mean here? It means these bacteria multiply outside our cells, so in spaces like your bloodstreams, your tissues, or on mucosal surfaces like your airways and intestines. When extracellular bacteria infect us, they cause disease mainly in two ways. First, they trigger inflammation. Inflammation, if you remember, is our body's response to infections, but too much of it can damage our tissues. So secondly, these bacteria produce toxins. They release substances like endotoxins, for example, the LPS from gram-negative bacteria, and exotoxins like in diphtheria or cholera toxins. So these toxins directly harm our cells or disrupt our normal bodily functions. So how does our immune system respond first? So firstly, through phagocytosis. Neutrophils and macrophages rush in to quickly engulf and destroy these extracellular bacteria. They can do this efficiently because these bacteria haven't adapted to survive inside our cells. These immune cells recognize bacteria using receptors like Mannose receptors, scavenger receptors, and complement receptors. And then they use phagolysosome to ingest and kill the bacteria. Another innate mechanism against extracellular bacteria is complement activation. So our complement proteins recognize bacterial surfaces and then trigger cascade pathways that helps our immune response. One result of that is opsonization, which means coating the bacteria to make it easier for phagocytes to grab and to ingest the bacteria. Another result is formation of the membrane attack complex or MAC, which punches holes and destroys the bacteria, especially those with thinner cell walls like Neisseria species. The next innate defense mechanisms is inflammation. Inflammation happens when dendritic cells and macrophages detect bacterial presence. So these cells then release cytokines, which are signaling molecules that increase vascular permeability, which means making blood vessels more leaky. So this allows leukocytes like neutrophils or monocytes to quickly move into infected tissues, and that clears out the localized bacteria efficiently. Now let's move from innate to adaptive immunity. So the key adaptive defense here is antibodies. Antibodies neutralize bacteria and their toxins by directly binding to them. They also opsonize bacteria, meaning marking them clearly for phagocytes to ingest. And also certain antibodies like IgG and IgM activate the classical complement pathway, which enhances bacterial clearance even further. Besides antibodies, our adaptive immune response involves helper T cells. So CD4 positive helper T cells coordinate the immune response by releasing different sorts of cytokines. Th17 helper cells specifically recruit neutrophils, which amplify inflammation at infection sites. Th1 cells produce cytokines like interferon gamma, which enhance the killing power of macrophages. And these helper T cells also support B cells to produce high affinity class switched antibodies. Now, what does that mean? Normally, B cells initially produce antibodies that are basic and not very effective. For example, they don't bind very tightly to pathogens. So helper T cells guide these B cells to upgrade the antibodies in two ways. So first, high affinity. High affinity means these upgraded antibodies bind more strongly and more precisely to their targets like bacteria, making the antibodies better at neutralizing or tagging the bacteria for destruction. And secondly, class switching, which means changing the type of antibody produced. Initially, B cells often produce IgM antibodies, which is good enough to start with, but not specialized. Helper T cells trigger them to switch to more specialized classes like IgG or IgA, and each of them better suited for specific jobs. So overall, with the help of T cells, antibodies become more stronger and more specialized, and that enhances our ability to fight infections. Now, some bacteria have an extra protection called capsules. This capsule is usually made of polysaccharides, and the capsules helps bacteria resist being eaten by our phagocytes. So to counter this encapsulated bacteria, our immune system produces critical antibodies, especially IgM and IgG2, without needing much help from T cells. So that's what it means by T independent. Another important point, our spleen plays a key role in clearing this encapsulated bacteria. So this is why people without a spleen are at higher risk of infections from encapsulated types of bacteria. Remember earlier I mentioned that sometimes our own immune response can harm us more than the bacteria themselves. So here's how it happens. When neutrophils and macrophages fight bacteria, they produce reactive oxygen species, ROS, and enzymes to destroy microbes. But these same molecules can unintentionally damage our own tissues if the response is excessive or prolonged. So too much inflammation can even lead to sepsis, which is a dangerous body-wide inflammatory reaction that can lead to shock. Now, another way bacteria can harm us is through something called superantigens. So on the top side here, you see how our immune system normally works. When a microbe infects us, only a small group of specific T cells recognize it. So these T cells have receptors, TCR, that match precisely with an antigen presented by MHC molecules on antigen-presenting cells. So because of this specificity, only those few T cells become activated and the rest are not. Now, superantigens work differently. A classic example is the superantigen called staphylococcal enterotoxin B, or SEB. So SEB binds to a specific region on T cell receptor called V beta 3 and connects it with MHC class 2 molecule. You see, superantigens, they don't bind inside the usual antigen binding groove here. So instead, they bind outside that groove. And that directly connects MHC class 2 molecules to many different T cell receptors. So this unusual connection activates a huge number of T cells at once, which is far more than a regular antigen ever would. So when so many T cells are activated simultaneously, they release massive amounts of cytokines, a phenomenon that we call cytokine storm. So this overwhelming cytokine storm leads to severe inflammation and symptoms like the toxic shock syndrome. Let's move to another issue our immune system sometimes causes, post-streptococcal sequelae. So what does sequelae mean? It means problems that happen after an infection is gone. Two classic examples you should know are rheumatic fever and post-streptococcal glomerulonephritis. In rheumatic fever, our body initially produces antibodies and T cells against streptococcal bacteria. After the bacteria is gone, those T cells mistakenly attack your heart tissues, and that causes heart inflammation. In post-streptococcal glomerulonephritis, our immunity kills the bacteria, right? What happens after that is our antibodies combine themselves with leftover bacterial components, basically the dead bodies of bacteria. And that combination forms immune complexes. And these immune complexes can deposit in your kidneys, causing damage to your kidney function. Notice that in both cases, it's not the bacteria that directly causes the damage. It's the immune system mistakenly attacking our own tissues after the infection is gone. Let's look at how extracellular bacteria evade our immune system. First, they use polysaccharide capsules. This helps blocks phagocytes from grabbing them and ingesting them. Second, some capsules contain sialic acid, a molecule that interferes with complement activation. And finally, extracellular bacteria can change or vary their surface proteins, especially on structures called pili. By constantly changing, they prevent our antibodies from effectively binding and neutralizing them. So all these strategies help bacteria survive longer inside our bodies. All right, that's extracellular bacteria. Next, let's shift our focus to intracellular bacteria. Intra means inside, so intracellular bacteria are the ones that manage to survive and replicate inside our own cells. Intracellular bacteria survive either inside phagosome, those compartments where our cells usually digest pathogens, or they escape into the cytosol, the fluid inside our cells. So by living inside our own cells, they hide from antibodies circulating outside our cells, in our blood and our tissue fluids. So this hiding strategy means our immune system has to use different approaches, like the cell-mediated immunity, to detect and eliminate these bacteria. Oh, we'll get there in a minute. Before that, let's see how our innate immune system responds against intracellular bacteria. So the innate defense starts with phagocytes like macrophages and neutrophils. They detect bacteria through special sensors called TLRs and NLRs, toll-like receptors and node-like receptors. Once detected, these phagocytes ingest and destroy the bacteria immediately. At the same time, natural killer cells, NK cells, respond early by releasing interferon gamma. So this cytokine boosts macrophages, making macrophages stronger at killing bacteria inside them. Another innate response involves the STING pathway. So this pathway detects bacterial DNA inside our cells, which triggers the production of type 1 interferons, mostly alpha and beta. Now let's move on to the adaptive immune response, specifically the role of Th1 helper T cells. When macrophages and dendritic cells detect intracellular bacteria, they release interleukin 12. This interleukin 12 pushes naive CD4 positive T cells to become Th1 cells. Th1 cells then release interferon gamma and express CD40 ligand. Both of these signals activate macrophages, turning them into stronger bacteria killers. Once activated, macrophages produce powerful molecules like nitric oxide, reactive oxygen species, and lysosomal enzymes, all of which effectively kill the bacteria inside phagosomes. But sometimes bacteria manage to escape from phagosomes into the cytosol, where they are safer from macrophages. When this happens, our immune system uses CD8 positive cytotoxic T lymphocytes, or CTLs, to handle it. So CTL specifically detects and kills our infected cells. So the infected cells have got to be sacrificed in order to destroy the bacteria hiding inside them. An example of bacteria handled this way is listeria, which escapes into the cytosol. Sometimes our immune system struggles to eliminate intracellular bacteria completely. When this happens, our body uses a strategy called granuloma formation. So you can think of granulomas as prison walls built by activated macrophages and Th1 cells. So they walls off the bacteria from spreading further. While granulomas help contain the infection, they come at a cost. Chronic activation of immune cells within granulomas can lead to tissue damage or necrosis, and also fibrosis, which is permanent scarring of our tissues. So this protective strategy explains why some diseases like tuberculosis, for example, can cause serious lung damage because of the granulomas rather than directly by the bacteria. Let's look at two clinical examples of intracellular bacterial infections, tuberculosis and leprosy. In tuberculosis, like I mentioned before, granulomas form primarily in the lungs, and these granulomas break down lung tissues, and that causes respiratory problems and lung disease. Leprosy actually gives a better example of how the type of immune response determines disease severity. So patients who mount a strong Th1 response develop the milder form of leprosy with fewer bacteria and limited tissue damage. In contrast, patients with a weaker Th1 and a stronger Th2 response get the severe form of leprosy with loads of bacteria and more extensive tissue destruction. So the type of immune response influences how bad the disease outcome is. Yeah, intracellular bacteria are clever survivors. They've evolved multiple strategies to evade our immune system. One common strategy is blocking phagolysosome fusion. This prevents the bacterial destruction inside macrophages. Another tactic is escaping entirely into the cytoplasm, so they avoid the deadly enzymes and reactive molecules inside the phagosome. They also detoxify or neutralize harmful substances produced by our immune cells, like reactive oxygen species and reactive nitrogen species. So these sophisticated evasion tactics explain why infections from intracellular bacteria can persist for years, becoming chronic and difficult to treat. All right, now let's leave our bacteria behind and look at viruses. Before we discuss immunity, let's quickly summarize how viruses cause disease. First, viruses often directly kill infected cells, a process called cytopathic lysis. Second, viral infections can trigger inflammation, causing additional tissue damage on top of that cytopathic lysis. Third, viruses can persist silently in our cells as latent infections. So they don't replicate actively but remain dormant and then reactivate later. So these different viral behaviors require specialized immune responses from our body. Our innate immune system reacts to viruses through type 1 interferons. So what happens is, first, the virus introduces its genetic material, like viral RNA, into the cell. Inside the cell, sensors such as TLR, RIG-I, or STING detect these viral molecules. And when triggered, these sensors tell the infected cells to produce interferons. Then these interferons spread out from the infected cells and bind to receptors on nearby uninfected cells. So once uninfected cells receive this interferon signal, they activate antiviral genes. And that activation expresses the synthesis of proteins that interfere with the virus's ability to replicate. So this entire response creates an antiviral state, which makes it difficult for viruses to infect new cells and replicate further. So I always imagine all of this like starships being invaded by alien pirates. So the first ship that got invaded sends a warning signal called interferons. So that signals protect the neighboring ships, the neighboring starships from pirate invasion. So the first invaded ship is gone, of course, but it helps the armada to win the battle against the pirates. Besides interferons, our innate immune system uses natural killer cells or NK cells as another critical defense against viruses. NK cells continuously patrol our body, so they go around checking cells for signs of viral infection. Normally, our healthy cells display MHC class 1 molecules, so think of these as ID cards that tell NK cells everything is fine. But some viruses shut down these MHC class 1 molecules. Why? Because remember from our previous lessons that cytotoxic T lymphocyte, CTL, require MHC class 1 to recognize and destroy infected cells. So the virus is trying to be clever to avoid cytotoxic T lymphocyte response. But that deception backfires with NK cells, because when NK cells detect this suspicious absence of MHC class 1 and or stress ligands on infected cells, they immediately respond by killing the infected cells and the viruses in them. So this rapid killing helps contain the viral infection during the early infection before the adaptive immune response fully kicks in. Now let's switch to the adaptive immune system and talk about neutralizing antibodies. Antibodies produced by B cells are crucial for fighting viruses when they are outside our cells. So the antibodies bind specifically to viral particles, blocking them from attaching to the surface of our cells and entering our cells. So this action is called neutralization. These antibodies also help prevent viruses from spreading between cells. And IgA antibodies are particularly important at mucosal surfaces, like in our respiratory tract, where they help block viruses at common entry points into our body. However, once viruses are inside our cells, antibodies alone are not effective anymore. So this is where cytotoxic T lymphocytes or CTLs or CD8 positive T cells step in. So CTLs constantly scan our cells for viral peptides presented by MHC class 1 molecules. So basically checking for viral fingerprints. When CTLs detect these viral peptides, they directly kill the infected cell, eliminating the viruses inside the infected cell. CTLs also release antiviral cytokines such as interferon gamma. So together, antibodies and CTLs form a highly effective combined strategy against viral infections outside and inside our cells. One remarkable feature of the adaptive immune system is memory. When our body encounters a virus for the first time, it produces long-lived plasma cells and memory T cells and B cells. So these cells stay quietly in our body, sometimes for years or even decades. They are ready to rapidly respond if we encounter the same virus again. So this is exactly why vaccination works. It exposes our immune system to harmless parts of viruses. So it trains our adaptive immune cells to respond quickly and strongly in the future. However, immunity is usually specific to viral serotype. So, for example, immunity to one influenza strain doesn't necessarily protect us from different influenza strains. Now, just like bacteria, sometimes our own immune response to viruses can unintentionally harm us. So, one way this happens is through CTLs. So, for example, in viral hepatitis, CTLs kill infected liver cells, and this immune reaction causes significant liver damage. Another scenario is a cytokine storm. Here, our immune system or our immune cells rather, release excessive cytokines, causing widespread inflammation, and those inflammation damages our own tissues. Viruses can also cause immune complex vasculitis. So this happens when antibodies and viral antigens form complexes that deposit in blood vessels. So they settle and build up in blood vessels. And that deposition causes inflammation and damage in those blood vessels. Viruses are notoriously good at evading our immune system. So one tactic is antigenic variation. So viruses frequently mutate, like influenza viruses do this a lot. They change their surface proteins, which helps them escape recognition by our immune system. They can also interfere with antigen presentation on MHC class 1 molecules, preventing detection by CTLs, cytotoxic T lymphocytes. Viruses can also suppress type 1 interferon pathways, which weakens our antiviral defense, and some viruses exhaust our immune cells. So, for instance, chronic viral infections can induce T cells to express inhibitory receptors like PD-1. For those who pay attention to my lesson on immunotolerance, for example, I mentioned that PD-1 reduces the T cells activity. In that context, it's good because it avoids autoimmunity. But in this case, it's bad. When viruses trigger it, it reduces the T cells' ability to clear the virus. All right, let's move on to our final group of pathogens, parasites. Unlike bacteria and viruses, parasites often have complex life cycles and they can involve multiple hosts. First, let's clarify the diversity of parasites we're dealing with. Protozoa are single-celled parasites, such as Plasmodium, which causes malaria. Helminths are larger, multicellular worms like roundworms and tapeworms. And then we have ectoparasites like ticks and mites that live on the external surfaces of our bodies. This wide diversity means our immune system needs a variety of specialized strategies to deal with each type. Let's start with innate immune defenses against parasites. Phagocytosis plays an important role against smaller protozoan parasites, although some protozoa can actually survive and even replicate within phagocytes. Against larger parasites, especially worms, eosinophils come into play. They release toxic granules that damage and weaken the parasite's outer surfaces. Complement activation also occurs in parasite infections, but as you learn later, many parasites have evolved various ways to resist complement attack. Next, let's look at how adaptive immunity deals specifically with protozoan parasites. Because many protozoa hide inside our cells, our immune system often relies heavily on Th1 helper T cells. These Th1 cells release interferon gamma, which activates macrophages to become better at killing intracellular parasites. And in addition, cytotoxic T lymphocytes, CTLs, play a crucial role, especially in infections like malaria, by targeting and killing infected liver cells. Antibodies also help neutralize protozoa, particularly when they are circulating in the bloodstreams. For helminths, because they are large and multicellular, they trigger a different adaptive immune response, typically dominated by Th2 cells. Th2 cells release cytokines such as interleukin 4 and interleukin 5. Interleukin 4 stimulates B cells to produce IgE antibodies. So these IgE antibodies bind to mast cells. So these antibodies prepare or prime the mast cells to react strongly when parasites are present. Interleukin 5 activates eosinophils, making them effective at attacking and damaging the worm's outer surfaces. But similar to intracellular bacteria, parasites can lead to granuloma formation, particularly in chronic infections. For example, schistosoma worms release eggs that are deposited in our liver. That triggers granulomas dominated by Th2 cells, which help contain these eggs. Think of granulomas as organized clusters of immune cells that gather around the eggs like a prison wall. However, these granulomas can cause tissue damage over time, leading to fibrosis. Another example is lymphatic filariasis. This is where worms lodge themselves in lymphatic vessels. Because the worms are there in the vessels, that causes persistent inflammation and fibrosis. And that fibrosis blocks the lymph drainage, meaning fluid cannot come out from the lymph. And that leads to severe swelling of the lymph. So while granulomas and fibrosis help control parasites, they also lead to chronic tissue damage and health problems. Parasites also have these interesting ways to evade our immune defenses. One common strategy is antigenic variation. The parasites frequently change their surface proteins. Trypanosome does this, and the constant changing of proteins makes it difficult for our immune system to recognize them consistently. Another strategy involves developing thick outer layers. We call it tegument in worms. These teguments protect them from complement proteins and even antibodies. Some parasites even hide within our cells or form protective cysts. Think of cyst like a tiny ball. So the cysts make the parasites invisible to our immune system. Yeah, this one is fascinating. Parasites also induce immune suppression. They encourage regulatory T cells activity or causes energy, which makes T cells unresponsive. It's like our enemy forces bribe our military officers to command our troops to stop attacking. Right? It's crazy that parasites learn to do this. So this is an example of the arms race between our immunity and pathogen. I wrote a philosophy of science paper about it actually, and I'll add the link to it in the description later in case you're interested. Okay, let's summarize the major points we have covered today. First, remember that our immune system uses distinct strategies depending on the type of pathogen: extracellular bacteria, intracellular bacteria, viruses, or parasites. Second, innate and adaptive immunity work closely together, so they complement each other to clear the infections. Third, while our immune responses protect us, they can sometimes unintentionally cause damage to our own tissues. And lastly, all these pathogens that we're talking about, bacteria, viruses, parasites, they have evolved sophisticated ways to evade our immune system, and that makes them persistent and that makes them challenging to control. That's all for today. Talk to you next time. Keep on reading, keep on learning. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.